into different groups. However, with the help of basic fundamental features such as level of organization, symmetry, cell organization, nature of coelom, segmentation, and notochord, scientists have classified animals into various groups. Let's understand each of these basic fundamental features in detail. Level of organization is the first basic fundamental feature that deals with the pattern of cell organization in an animal. There are 4 patterns of cell organization: cellular, tissue, organ, and organ system. Sponges exhibit a cellular level of organization where cells are loosely arranged. Tissue level of organization is more complex and is displayed in coelenterates. Here, the cells that perform the same function are arranged into tissues. Organ level of organization is exhibited in members of platyhelminthes and other higher phyla. Here, the tissues are grouped to form organs, and each organ performs a particular function. The last cell pattern is the organ system level of organization. Here, the organs are associated with one another to form a functional system, where each system performs a specific physiological function. Animals like annelids, arthropods, mollusks, echinoderms, and chordates exhibit this level of organization. Organ systems can also differ in complexity in various animal groups. For example, platyhelminthes have an incomplete digestive system, where a single opening outside the body serves as both the mouth and the anus, whereas humans have a complete digestive system that has 2 openings, a mouth and an anus. You will also find 2 types of circulatory systems in animals: open and closed. Symmetry is another feature by which animals are classified. They can be asymmetrical, like sponges, or have radial symmetry, like coelenterates, ctenophores, and echinoderms. However, most animals like annelids and arthropods have bilateral symmetry. Cell organization is the next feature used for classification. There are 2 types of cell organization: diploblastic and triploblastic. Coelenterates have diploblastic organization, where the germinal layers of the cell consist of an external ectoderm and an internal endoderm. Members from phylum Platyhelminthes to chordates have triploblastic organization, where a third germinal layer, the mesoderm, is found between the ectoderm and the endoderm. The fourth important feature by which animals are classified is the nature of coelom. The coelom is the body cavity lined by mesoderm. The presence or absence of this cavity helps classify animals. There are 3 categories: coelomates, acoelomates, and pseudocoelomates. Coelomates are animals that possess a body cavity, such as annelids, mollusks, arthropods, echinoderms, hemichordates, and chordates. Acoelomates are animals in which the body cavity is absent, such as platyhelminthes. Pseudocoelomates are animals that possess a pseudocoelom, such as askhelminthes. A pseudocoelom is a body cavity in which the mesoderm is not lined, but rather scattered between the ectoderm and the endoderm. Some animals are also classified on the basis of segmentation. The body of some animals can be divided externally and internally into segments. Sometimes, the segments in animals have serial repetition of some organs, which is seen in earthworms. This phenomenon is known as metamerism. One of the most important features for animal classification is the presence or absence of a notochord. Some animals develop a rod-like structure on the dorsal side during embryonic development, which is known as the notochord. Animals with a notochord are called chordates, while animals without a notochord are called non-chordates. Members of phylum Porifera to echinoderms are non-chordates. We just studied how we can classify animals into different categories on the basis of some fundamental features. Animals belonging from phylum Porifera to phylum Echinodermata are categorized as non-chordates. Non-chordates are animals without a notochord. Let's take a look at the important characteristic features of the phylum Porifera to the phylum Aschelminthes. The first phylum under non-chordates is Porifera. The members of this phylum are known as sponges. These are primitive, multicellular, asymmetrical, marine animals with cellular level of organization. The body of sponges is supported by a skeleton made up of spicules or spongin fibers. Sponges have a water transport or canal system which helps in food gathering, respiratory exchange, and waste removal. In this system, water enters through minute pores called the ostia into a central cavity called the spongiocele and comes out through the osculum. The spongiocele and the canals are lined with special cells called choanocytes or collar cells that help in the ingestion of food particles followed by intracellular digestion. Sponges are hermaphrodite and they reproduce asexually by fragmentation as well as sexually by formation of gametes. In sponges, fertilization is internal with indirect development. Cycon, spongula, and euplectula are some common examples of sponges. The second phylum is Coelenterata. The members of this phylum are aquatic, mostly marine, sessile or free-swimming, radially symmetrical, dipoblastic animals with tissue level of organization. Some coelenterids, like corals, have a skeletal structure made of calcium carbonate. Coelenterids have cnidoblasts on their tentacles and body, and hence this phylum is also known as Cnidaria. Cnidoblasts are used for anchorage, defense, and to capture prey. Coelenterids have a central gastrovascular cavity with a single opening called the hyperstome. Digestion in them is both extracellular and intracellular. Coelenterids exhibit two basic body forms, polyp and medusa. Polyp is a sessile and cylindrical form like hydra and